Hi, I'm Janine Cody, and this is the second of 10 short videos addressing some of the frequently asked questions that I get about chromosome 18 abnormalities. And then the first video, we talked about what is a chromosome variation or a chromosome abnormality, if you want to use that term, and found out that now we know they can be very, very, very small. So the question becomes, how common are chromosome variations? And just to review, here is a picture of human chromosomes from a blood cell taken under a microscope and then cut apart and lined up so you can see all the pairs of chromosomes. As a reminder, one's from mom, one's from dad. And a chromosome change, a chromosome abnormality, means having a duplication or deletion of one of the two copies of the chromosome. So the other one is intact and presumably perfectly normal. And when chromosome abnormalities were first discovered to be a cause of human variation, human disease, or human conditions, this was the technology that was used. And so in those days, you couldn't detect anything very small, as you'll know from the previous video. And so in assessment of many children born and randomly looking at their chromosomes, it was determined that about 1 in every 200 babies born had a chromosome change or a chromosome abnormality using that technique. But of course now, thanks to the Human Genome Project, we can detect very, 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 very small deletions or duplications. And in this picture, we have a picture of chromosome 18, and it just shows the black and white bands from a high-resolution karyotype or microscopic image. And the red box shows the smallest detectable deletion or duplication. That red box could be anywhere around the chromosome, but here I just have it at the end for this illustration. So that would be pretty big by molecular terms, and here's what we know now. In the box below that, it shows a zoomed-in view of what is contained there, and at the top part of the lower box, you can see a bar that indicates the scale of 2 megabases. So that's 2 million base pairs of DNA. That's how big it would be in this image. Below that is the actual scale with the position of the base pairs from about 75,000 to 78 million to 75 million to 78 million. Below that is the important part. Below that is the position of the genes in that region. So each one of those little vertical bars, some connected by a horizontal bar, shows the position of a gene. Next to it is the acronym for their name. They're just scattered down that image so that it's readable because the names are kind of in the way. But in actuality, they are one after the other. So you can see at the end, on the right-hand side, at the end of the chromosome, they're clustered. Then there's a big gap there. And so this is, the scale of today, and we could actually determine, even missing one or duplicating one base pair of DNA. So that's not really the analysis anyone really has done in real life in a clinic. And so if you look for changes in copy number, deletions or duplications on one of the two copies of a chromosome that are 500,000 base pairs or bigger, and just to point out here, 500,000 base pairs could include 3 genes, like shown here. It could be to the right a little bit, include no genes, or it could be at the end here and include 4 or 5 or 6 genes. So 500,000 base pairs may or may not include genes, and it doesn't really even tell you how many genes might be included. But if a large number of babies are randomly checked for deletions or duplications of this size or larger, this is what we find. We find that 5 out of every 200 babies has a new chromosome change of that size or larger. And these are new changes that are not found in their parents. So, how many is that really? Well, I live in San Antonio in Bexar County, and that would mean that 664 babies every single year are born in Bexar County with a chromosome change of 500,000 base pairs or bigger that is new in them, that they did not inherit from their parents. So now I know you're thinking, okay, but their parents, they inherited some of those new ones, and so what if you include the ones they inherited from their parents, who may have inherited some from the grandparents, etc.? So when you look at that, all the changes that are new or inherited in 200 babies, then that reveals that everybody has chromosome abnormality. In fact, if you look at base pair changes of 100,000 base pairs or bigger, on average, each one of us has 10 of those. And if you go even smaller to 50 base pairs, which is the new definition of a chromosome change, a chromosome variation, that we each have at least 50 of those on average. So chromosome variation is really, really common. The number of base pairs in any one person's genome is going to vary a lot. So what does this mean? Well, we're back to, of course, the slide I use all the time. Anything you can do to this picture of chromosomes with scissors, glue, and a copy machine can really happen, and it does really happen. And what that means is, we all have chromosome abnormalities, every single one of us, and we even have more than one. But most of us, or many of us, are relatively healthy, and so how do we know which ones affect health and which ones don't? And so that is the million-dollar question that we're going to start to address in the next 8 of these videos. So stay tuned.